A very good morning. Welcome to my oral presentation. As we know that arsenic is considered one of the most dangerous pollutant in the surrounding due to its prevalence in the soil and water, and it has also an ability to accumulate in the food chain, which causes the several diseases in the human. To overcome this problem, scientists have designed the whole cell biosensor based on GFP in order to detecting the arsenic concentration in the environment. We have construct this particular gene construct which contains GFP muta3A gene, encodes for GFP. We use plasmid pad and Bacillus subtilis as a reporter gene fusion act as a carrier. So which has an innate ability to detect the arsenic from the surrounding. In the next slide, I have shown the materials and methods and which we use in this particular experiment. We use Bacillus subtilis medium, which is basically the lactonic broth is used, and incubator for the incubation of the cell culture. And in order to measure the optical density, we use spectrophotometer. We also prepared the stock solution for in future for serial dilution. In this and this slide shows how I design the vector and ensuring the vector. So here I took the DNA fragment which contained the operator or a promoter region of the ARS operon. This particular DNA template we amplified in the PCR, and we also added the forward reverse primer in order to get the specificity. Once we get the amplified DNA, we insert it into the PJET blood vector, which act as an intermediate for preparation and verification of DNA, then integrated into the EcoRN BAM-H1 site of this plasmid, and we get the gene construct which named as PAD PRS GFP muta3A. In order to define the verification of particular DNA template, we added a specific primer. So this is how the verification is done. Once the verification is done, we added into the E. coli for the replication, inserted this particular gene interest construct. This particular gene interest further transformed into the Bacillus subtilis in order to get the strains Vimago 49, which act as a biosensor. In order to detect the how this Bacillus subtilis strains get exposure in the arsenite, we take an adequate from the Bacillus culture, incubate for 12 hours, and 1 ml of from each subculture is pelleted by centrifugation and washed and resuspended in PBS. We prepare the stock solution for serial dilution. Once it done, we diluted this subculture, spread on LB medium, again incubated for overnight. And here the result, which shows that whole cell biosensor has a great effect in detecting the noxious effect of arsenite. In the next slide, I show how this particular gene construct inserted and its mechanism. So this ARS operon has a four gene, ARSR, RSB, RC, and YQCK, and the green arrows show the operon promoter responsible for the transcription. This particular DNA contains ARS operon inserted in this pad, and then we have also inserted GFP3A gene in the upstream of the regulatory region of this pad plasmid. Then we transform this gene construct into the Bacillus subtilis. And here is a mechanism that when this particular bacteria come in contact with the arsenite, the ARS regulatory protein encoded by ARSR get inactivated, which then responsible, and once it get inactivated, it is responsible for further um, uh, uh, re- expression of these other gene responsible to detect the arsenite in the surrounding. And simultaneously, the GFP muta3A gene show the fluorescency which act as a biosensor. This is how we can able to detect the arsenite in the surrounding, the concentration of the arsenite in surrounding. Thank you so much.